morning, I am Madhurima Datta, Zoology and Biology educational content creator. Now's content for B.Sc and M.Sc Zoology Honours, Physiology Honours, Microbiology Honours, Botany Honours, Biochemistry Honours and Molecular Biology Honours. An exam for CUT PG exam, GATE exam, DBT exam, IIT JAM exam and CSI NET Life Sciences exams. Paper Molecular Biology, Unit 1, Introduction of the Molecular Biology, and today I will discuss about the properties of DNA. Let's go. Chemical properties and chemical bonds of the DNA. First is sugar phosphate backbone. Phosphodiester linkage links the 3' OH of 1, 2' deoxyribose to the 5' OH of the adjacent sugar. And the negative charge on the phosphate group at physiological pH or pKa equals to 1, which makes DNA highly polyanionic, means negative charged. Next, base stacking and hydrogen bonding. That is first canonical base pairing or Chargaff's rule. Adenine pairs with thymine by 2 hydrogen bonds and guanine pairs with cytosine by 3 hydrogen bonds. Next, stabilization force. Base stacking interactions via van der Waals forces and hydrophobic interactions between aromatic rings account for most of the structural thermodynamic stability, which is exceeding the contribution of H bonds or hydrogen bonds. Next, spectroscopic and thermal dynamics. First characteristics is UV absorption peak. Nucleotide bases strongly absorb UV light at lambda equals to 260 nanometer due to pi to pi prime transition in their conjugated ring systems. Properties is hyperchromic effect. Unstacking of bases during denaturation increases UV absorbance at 260 nanometer by roughly 30 to 40%. Next property is melting temperature. Temperature at which 50% of the helical structure becomes single-stranded, which is driven higher by high GC content, means 3 hydrogen bonds plus stronger base stacking energy and higher salt concentration. Salt concentration means cations neutralize phosphate backbone repulsion. Next property is Cott curve analysis. Measure renaturation kinetics provides insight into the genome complexity sequence, redundancy and size. Next, chemical modification and damage mechanics. Post-replicative chemical attraction affect gene expression patterns and genome integrity without altering the primary base sequences. Next, epigenetic modifications. First is 5-methylcytosine, which is added by DNA methyl transferases at CpG island. It projects into the major group, inhibiting transcription factor binding or recruiting methyl CpG binding domain and also called MBD domain proteins to repress the gene expression. Next, cytosine demethylation pathway. Oxidation of 5-MC by TET enzymes, that is 10-11 translocation, which is yields by 5-hydroxymethylcytosine, 5-formylcytosine and 5-carboxylcytosine, leading to active or passive demethylation. Next, common DNA damage reaction. First is depurination, cleavage of the N-glycosidic bond, which is leaving an apurinic or apyrimidinic site, that is same 10,000 event cell per day. Next, deamination. Cytosine deaminates spontaneously to uracil and adenine to hypoxanthine and guanine to xanthine. This mechanism I will discuss the next slides in briefly. Next, UV radiation lesions, which induces covalent cross-linking between adjacent pyrimidines, producing cyclobutane pyrimidine dimers and 6 to 4 photoproducts. Next, non-covalent interactions and intercalation dynamics. Small molecules and proteins recognize DNA through specific binding geometries that modify its local physical parameters. First is minor groove binding. Ligands, that is crescent-shaped molecule like netropsin, distamycin and hoexest, which is molecular weight is 33,258 Dalton. Mechanism bindly preferentially to AT rich region driven by electrostatic interaction, van der Waals contact and water displacement entropy. They fit into the narrow minor groove without disrupting base pairing. Next, intercalation. Ligands. Planar aromatic ring systems such as ethidium bromide and propidium iodide and doxorubicin. Mechanism: slip between the adjacent base pairs via hydrophobic pi stacking interaction, and structural consequence: unwinds the local helix by SIM26 and circ for ETBR, elongates the DNA contour length and stiffens the backbone and alters the supercoiling density to positive supercoils and high concentration. Next, a short note, the nucleosome core particle. In the next slide, I will discuss about the brief notes about nucleosome. First is the composition. An octamer of the core histone, it is 2 times H2A and H2B dimer and 1 times H3 and H4 2 tetramer wrapped by 147 BP of bDNA in 1.65 left-handed superhelical structure. Next, interaction. DNA histone binding is largely sequence independent, driven by 14 salt bridge between the positively charged basic lysine and arginine residue on histones and the polyanionic phosphate backbone. Next, histone tail modification. Flexible N-terminal tails projecting outside the core undergoes post-translational modifications or PTMs. Next, acetylation, that is H3K9AC and H3K27AC, which neutralizes positive charges on lysine disrupting backbone tail electrostatic attraction, loosening chromatin into accessible euchromatin. Next, methylation, which is acts as structural docking sites for effector protein without directly altering charge. H3K4 methyl 3 marks active transcription, whereas H3K9 methyl 3 recruits HP1, means heterochromatin protein 1, to encodes DNA into heterochromatin. Now the topic has ended. For all video of B.Sc and M.Sc in Life Science, Biotechnology, Zoology, Biology, please subscribe my YouTube channel Biogenesis by Madhurima and follow my Facebook account Madhurima Datta. Thank you everyone.